In this lecture, we are going to do some exercises of the t-test, and in the previous lectures, we had discussed that what are the different types of the t-test and what are their specific requirements and what are their common requirements. And there are basically two types of the t-test that we studied. The student's t-test, which is the t-test for the independent means or the unmatched or the unpaired observations, and we studied the paired t-test, which is the t-test for dependent means or the matched observations. And we saw their general requirements, that what are the requirements for what are the requirements for these t-tests to be performed. And then we saw that how these two types of the t-tests they are executed, and we solved a couple of exercises over there for each of the tests. And we also discussed that how these t-tests can be performed on Microsoft Excel. So today we are going to do some further exercises so that you have a clear concept and differentiation of the two types of the t-test that we perform, that what are the conditions under which we have to perform the t-test for the independent means, and what are the conditions under which we have to perform the t-test for dependent means. So here we are with the exercise number 1 for today, and this exercise is about the plasma ACE levels. And ACE is the angiotensin converting enzyme, which is a very important enzyme found in animals. And this enzyme is involved in is part of the renin-angiotensin system, and this is involved in certain metabolic functions of the body, including the kidney functions and the blood pressure regulation. So this is a very important enzyme, and this is the exercise about the plasma ACE levels that were measured among healthy and hypertensive mice. And the unit of their measurement is picogram per deciliter here. So find out if there is a significant difference in the mean plasma ACE levels among healthy and hypertensive mice. So what kind of the t-test are we going to do? This is obviously we are going to apply a t-test because this is the comparison of mean values or the comparison of averages. So the question is that what kind of t-test are we going to use? Are we going to use the matched observations t-test or the unmatched observations t-test? So in this case we have two groups of mice, healthy mice and hypertensive mice, and the variable is the plasma ACE levels, and this variable is measured only once from each of the units in the two groups. So therefore these observations are independent, and we are going to apply the student's t-test, which is the t-test for independent means. So this is the data. These are the observations that we have. So these are the plasma ACE levels, and the green boxes or the green columns, they show the plasma ACE levels in healthy mice, and the blue boxes or the blue columns, they show the plasma ACE levels in the hypertensive mice. And these pink boxes, they show the number of mice, right? So this is just the serial number of mice. So here you can see that this is one group of mice. So in this group of mice we have 20 mice, and we have taken 20 observations. So one observation from each mouse. And what are these observations? These are the plasma ACE levels. So this constitutes one group, and these are 20 healthy mice. Now the next group is of the hypertensive mice, and these mice are also 20 in number, and we have 20 observations from these mice of the same variable, which is the plasma ACE levels, right? So I think the concept is clear to you, that what is the question and what kind of data do we have. Now this is the descriptive statistics for this analysis, for this exercise. So the number of healthy mice is 20, and the number of hypertensive mice is also 20. And the mean plasma ACE levels in healthy mice are 15.76 picograms per deciliter, and the plasma ACE levels in the hypertensive mice are 20.23 picograms per deciliter. And then the standard deviation of the plasma ACE levels from the mean value in the healthy mice is 2.669 picograms per deciliter, and in the hypertensive mice this is 3.2677 picograms per deciliter. And then in the healthy mice the variance is 7.1225 picograms per deciliter, and in the hypertensive mice this is 10.678 picograms per deciliter. So this is the descriptive statistics. Now what you have to do is that you have to assign numbers to these samples yourself. So I haven't assigned the numbers, that what is sample number 1 and what is sample number 2. So you have to assign these numbers yourself, that what you are going to label as sample number 1 and what you're going to label as sample number 2. So these are the two samples, or these are the two groups that you have. So this is our first exercise. Now we have the second exercise, and this exercise is about the same variable, and which is the plasma ACE levels. So in this case, the plasma ACE levels were measured among hypertensive mice before and after treatment with a drug. So in this case, we have only one type of the mice, one group of the mice, which are hypertensive mice. But in this case, again, the observations are different. So these observations are taken twice from each of these mouse. And these observations are taken before the treatment and after the treatment with a drug. So these are the matched observations. The observations are dependent, and these observations are present in pairs, and a pair of observations is taken from each mouse. So these are repeated measures. So we are going to apply the t-test for matched observations in this case. So the question is, find out if there is a significant difference between the mean plasma ACE levels of mice before and after treatment of the drug. So this is our state of affairs. Now these are the observations that we have, and in the green boxes we have the observations of the plasma ACE levels before treatment, and in the blue boxes we have the plasma ACE levels after treatment. And in the pink boxes we have the serial number of mice. So you can see that there are 40 observations, but these observations are taken from 20 mice, which means that from each mouse we are getting a pair of observations, and observations are being repeatedly measured. So 15.4 and 8.4 is the first pair of observations that we have from mouse number 1, and so on. So we have a pair of observations from each mouse. So this is these are first 10 mice, the pairs of observations from the first 10 mice, and these are the pairs of observations from the next 10 mice. But are these two types of the mice, are these two separate groups? No, they are a single group, right? They are a single group, but the observations are repeated. The observations are matched, and these are the repeated measures observation from the same group of mice, right? So we are going to apply the t-test for the matched observations here.